So, good morning all. Let's have a revision on renal regulation of acid-base balance. We know the normal pH of plasma is 7.4. So, in normal life the plasma pH used to vary, means it is it's not vary, but it will be in a very narrow range, that is 7.38 to 7.42. So, the pH of interstitial fluid is generally 0.5 units below that of the plasma, and the pH is below the normal pH, that is 7.38. It is we know it is acidosis, and life is dangerous this pH when the pH is lowered below 7.25. And acidosis, which can lead to CNS depression and coma. Death occurs when the pH is below 7. And if the pH is more than 7.42, it is known as alkalosis. That also dangerous, and it can lead to neuromuscular hyperexcitability, tetany, etc. And even death occurs when the pH is above 7.6. And now our topic is how the regulation of acid-base balance occurs. There are three main things, or three lines of defense. The first line is blood buffers, that we have already been discussed. Then second line, respiratory regulation, and the third line of defense is renal regulation. That is what we are going to discuss. So, the normal urine pH is around 6, and the lowered pH is due to extracellular, that is lower than that of the extracellular fluid. This is called acidification of urine. The pH of urine which vary from low such as 4.5 to as high as 9.8 depending on the amount of acid excreted. Now, let us go in detail about the renal regulation of acid-base balance. It is mainly done with four mechanisms. First one is excretion of hydrogen ions, second reabsorption of bicarbonate, third excretion of titrable acids, that is net acid excretion, and the fourth one is excretion of ammonium ions. These four can be explained with the four diagrams that I will be showing now. So, the first one is excretion of hydrogen or generation of bicarbonate, or it is known as the alkali reserve. It occurs in PCT, proximal convoluted tubules. Here the carbon dioxide, okay, let us see in figure. This figure you must draw, which shows the excretion of hydrogen ions in the PCT. So, in PCT, proximal convoluted cells, carbon dioxide combines with water in presence of carbonic anhydrase enzyme. It leads to the formation of carbonic acid. This carbonic acid in PCT itself broke down into bicarbonate and hydrogen ions. Here, this hydrogen ion in exchange with the sodium in tubular lumen, this hydrogen ion will move to tubular lumen where hydrogen ions is excreted, and from tubular lumen sodium will be absorbed to the PCT and which will be enters into the plasma. So, again in PCT the bicarbonate which is broken down from the carbonic acid also which reaches to the plasma, and the sodium and bicarbonate will be combined to form sodium bicarbonate, and here the bicarbonate alkali is recovered to plasma. So, this is known as alkali reserve and excretion of hydrogen ions. Second thing, reabsorption of bicarbonate. This is mainly as a mechanism to conserve a base. There is no net excretion of hydrogen ions. So, here in this figure again, tubular cell, the carbonic acid synthesis already it is done with the carbonic anhydrase enzyme we have already seen here, which explains the hydrogen. Previously we have seen the hydrogen excretion. Instead of that, here we are focusing on the bicarbonate ion. This bicarbonate which moves to plasma, and that alkali is recovered to plasma. Okay. And here hydrogen, when it is reaches to the tubular lumen, this hydrogen can combine with bicarbonate which is already present in the lumen, that is already synthesized from the sodium bicarbonate breaking down into sodium and bicarbonate. Sodium will move to the tubular cell and this bicarbonate will combine with the hydrogen in tubular lumen and form this carbonic acid. And the enzyme carbonic anhydrase present in the tubular lumen helps to breaking down the carbonic acid into carbon dioxide and water. This will reach us to the tubular cells. So, in the second figure which explains the reabsorption of bicarbonate from the tubular fluid. So, here bicarbonate is reabsorbed from the tubular fluid. And the third is excretion of hydrogen as titrable acids. Here it occurs in the DCT, distal convoluted tubule, net acid excretion occurs. The hydrogen ions are generated in the tubular cell by carbonic anhydrase. Bicarbonate generated within the cell passes to the plasma, and the term titrable acidity of urine refers to the number of milliliters of N/10 sodium hydroxide which required to titrate 1 liter of urine to pH 7.4. This is a measure of net acid excretion by the kidney. So, you should write the definition of what is the meaning of titration of titrable acidity of acids. Okay, here the same thing, tubular cell, carbonic acid breaking down to bicarbonate and hydrogen. Hydrogen is excreted tubular lumen which will be combined with sodium biphosphate to form monosodium hydrogen phosphate, and this monosodium hydrogen phosphate is excreted, and this is which explains the phosphate mechanism in tubules which help for the acid base balance. The acidification of urine in the form of sodium biphosphate. And the last one, excretion of ammonium ions which predominantly occurs in the DCT. So, remember the first two, that is excretion of hydrogen ions and the reabsorption of bicarbonate which occurs in the proximal convoluted tubule, and the last two, that is excretion of ammonium ions and the titrable acidity or excretion of acid which occurs in the DCT. Excretion of ammonium ions help to excrete hydrogen and reabsorb bicarbonate. This mechanism also helps to trap the hydrogen ions in the urine so that large quantity of acid may be excreted with minor changes in pH. The excretion of ammonia helps in elimination of hydrogen ions without appreciable change in the pH of the urine. This figure which explains the ammonia mechanism, you can see the same thing, carbon dioxide plus water in presence of carbonic anhydrase to carbonic acid, break down to bicarbonate and hydrogen in tubular cell. Bicarbonate will be reabsorbed to tubular fluid, that is plasma, and hydrogen will be moved in exchange with the sodium. This hydrogen will combine with the ammonium. This ammonium, here we are focusing on how this ammonium reaches to the tubular lumen, that is from the tubular lumen glutamine, the amino acid glutamine hydrolyzed by the glutaminase enzyme in presence of water to form glutamic acid and ammonium, that is deamination of glutamine done with glutaminase, and this ammonia reaches to the tubular lumen. This will combine with the hydrogen ion synthesized from the breaking down of carbonic acid, and this ammonia combined with the hydrogen to form ammonium ion, that is NH4+, and hydrogen is trapped and will be excreted.